afternoon, everyone, and welcome to the front row. My name is Jamie Williamson. I'm a professor here at Scripps Research, and I'll be your host today. And it's our pleasure to have Luke Lairson. The subject of Luke's lecture is going to be multiple sclerosis. So this is an autoimmune condition. So it's a terrible condition where your body's immune system actually attacks your nerve cells and leads to a degradation of function. And it's typically a progressive disease. And so I think the question that Luke's trying to answer is, can we stall this process or possibly even restore the function? And this is done by trying to activate some endogenous cells called stem cells in your body, and it's their job to sort of repair the neurons over your lifetime and keep them in good shape. They're kind of the highway crews of your nervous system. And so if you can activate these stem cells, then you can actually hopefully improve the function of the nervous system. So that's regenerative medicine, trying to use the body's own capabilities to repair a disease state. Now, Luke is a member of the chemistry department, but he's the hallmark of an interdisciplinary science. So he can make molecules, but he's got neurons growing in his lab, and he's got neurons in his brain too. But he's got neurons growing, and they have the capability of testing compounds and trying to understand this process of regeneration of the myelin sheaths in your neurons that is the root cause of MS. So Scripps is a not-for-profit institute. We get lots of funding from the federal government. We also count on support from the community, and there's actually a table outside afterwards, the gold table, and you can learn more about Scripps and ways you can interact with Scripps and support Scripps if you're so inclined. But the primary goal of today's lecture is it's our outward-facing component to let the general public know what's going on here at Scripps. And so I'm going to let Luke introduce himself a little bit, but at the end, I'm going to come back and we'll have a little discussion. So we have about 200 people here in person. We have about 4,000 registered to watch online, and Zoom willing. If maybe you don't know, but there's a global Zoom crisis today, and I think it might be a bit of a challenge, but if there is a Zoom crash, we'll have the video available by the next day, so you can catch up where you left off. So if the Zoom holds, you can type a question in, and it'll get relayed to me, and we can have a discussion. If you're in the audience, you can raise your hand, and someone will attend to you, and we'll have a little discussion. I'm sure there's going to be lots of questions. So Luke's going to introduce himself, but let me just say he was recently promoted to tenure and full professor in the chemistry department. So on that note, welcome. Yeah.
All right. Okay, thank you very much, Jamie, and thank you everyone for being here, both in person and hopefully online. It really is a pleasure and a privilege for me to be here today and be able to speak to this audience about some of the work going on in our lab in the context of remyelination and MS. Jamie did throw me a curveball just as I was about to get up here, telling me I had to introduce myself to you, which people who know me know that this is not my favorite thing to do, but I'll give it a quick run through. So I grew up in a very small town in rural Ontario, Canada, and then I did my graduate work in Vancouver, British Columbia, studying enzyme mechanisms and working with a really incredible scientist named Steve Withers. In there, I learned how to make molecules, and the objective was to use and design molecules to specifically impact a biological function. And towards the end of my graduate career, I decided I wanted to take an opposite approach, and I wanted to use nature's response to structure to guide hypothesis. And this is an approach called phenotypic screening, which forms the basis of a discipline called chemical biology. And Scripps is really a mecca for that entire field of study. So around 15 years ago, I came to Scripps to do my postdoc with Pete Schultz, who I'm sure many of you know, who's the president and CEO of Scripps. And I have been here ever since. And so the story I'm going to tell you about today started shortly after my postdoc here at Scripps, and all of the findings we made were made using this approach called phenotypic discovery. Okay, let's get started with the actual seminar. So just to give you an idea of where we're going and the topics I want to cover today, I'd like to start by telling you about some recent developments in our understanding of the molecular basis for MS disease, and how these insights have contributed to recent breakthrough therapies that are highly effective in the context of treating relapsing, remitting forms of MS. And in the second part of the talk, I'll tell you about a regenerative process called remyelination, which holds tremendous promise in the context of developing therapeutics for progressive phases of MS. And one of the things I hope to be able to relay to you, and something that you come away from today's lectures with, is the incredible and essential role that academic science plays in the discovery and development of new medicines. And everything I'm about to tell you was done by academic researchers, both academic clinician scientists and research scientists in academic settings. Okay, so let's get into it. MS is a devastating autoimmune disease that impacts the central nervous system, so the brain and spinal cord of a significant number of people around the world, disproportionately so in countries like the United States and Canada, as well as countries in Northern and Western Europe. And the reason for that is rates of MS increase as you move geographically away from the equator. And so, well, it's estimated that the number of cases of MS globally is around 3 million. In the United States alone, there's 1 million cases of MS. And what that equates to in the states, the northern states of this country and in Canada, the rate of MS is 1 in 400 people. So it's a highly prevalent, devastating disease that impacts a significant number of people. As you may also be aware, like many autoimmune disease, MS is a disease that disproportionately impacts women. So women are around 3 times more likely to be diagnosed and develop MS disease. MS is characterized by an adaptive immune response to a specialized cell type present within the central nervous system called an oligodendrocyte. And the neurological symptoms that arise in MS disease are the result of a process called the demyelination of the axons of neurons within your brain and spinal cord. And so what are these oligodendrocytes and myelin? Well, as you may or may not be aware, there are multiple cell types present within your brain and spinal cord. In addition to the relatively boring old neurons that you are more than likely aware of, there are multiple types of glial cells present, which play equally important and essential functions in the maintenance and ability of your central nervous system to function. This includes astrocytes, microglia, and oligodendrocytes. And it's these oligodendrocytes that we'll be talking about in some detail today. These cells associate with and wrap the axons of neurons with a lipid-rich myelin sheath, which is required for the function and survival of neurons. And so these lipid-rich myelin sheaths insulate the axon, which facilitates saltatory conduction, which is the function of a neuron. And these myelin sheaths also provide metabolic support, which is essential for the survival of neurons. So these cells play incredibly important roles in both the maintenance and function of your nervous system. And this is a nice cartoon representation of these cells. Of course, these are real things. So for example, this is an image, immunofluorescent analysis of an oligodendrocyte that we generated in the lab using a stem cell population that we isolate from rat optic nerve. And you can quantify these essential myelin sheaths using techniques like electron microscopy in the context of a living organism, which allows you to interrogate how mechanisms impact oligodendrocyte function and myelination. And we'll be talking about this in some detail in the second part of the talk. As I mentioned, MS is an autoimmune disease. It's characterized by adaptive immune responses specifically to these oligodendrocytes. So cells of your adaptive immune system, like T cells and B cells, specifically recognize and target these oligodendrocytes, leading to their cell death. And the loss of these oligodendrocytes leads to the loss of these essential myelin sheaths through a process called demyelination. And in the absence of repair, the loss of these myelin sheaths ultimately leads to the loss of these axons and the death of neurons, which leads to permanent neurological dysfunction, which is associated with progressive forms of MS. In the time since I started working in this area, there's been significant breakthroughs in the development of effective therapies for relapsing remitting form of disease, all exclusively which target inflammatory or immune-related mechanisms. The discovery and approval of a drug at Novartis called fingolimod, which targets a receptor called S1P receptor, really represented a huge breakthrough in the field, and this was really the first orally available effective therapy for MS. Our own Hugh Rosen, working with Ed Roberts here at Scripps, developed a second-generation drug targeting this same receptor that had improved receptor subtype selectivity, which significantly enhanced the safety profile of this drug. And in 2020, this drug was approved for the treatment of relapsing remitting MS, as well as other inflammatory and autoimmune-related indications. And across the street, when Pete was running GNF, a drug called BAF312 was developed, and this now represents the frontline approved therapy that Novartis uses targeting this mechanism, which is approved for use not only in relapsing remitting MS, but also in the context of secondary progressive MS, where there's small but statistically significant impacts on disease progression. More recently, it's been demonstrated that targeting B cells is a highly effective way to address this disease. And so these are really exciting new results. So B cells are another really powerful arm of the adaptive immune response. These cells give rise to the plasma cells that produce the antibodies that facilitate antibody-mediated responses. They also produce pro-inflammatory cytokines. And probably most relevant in the context of MS disease, these cells can act as antigen-presenting cells, which serve to prime T cell response, which leads to, presumably in the context of MS, T cell-mediated destruction of oligodendrocytes. This work was spearheaded or led by an academic clinician scientist named Stephen Hauser up the coast at UCSF, who really had the insight and volition to test this hypothesis in the clinic that B cells played a driving role in the progression of MS disease. And what the results of his phase 3 clinical trials clearly demonstrated was that by depleting B cells in MS patients, there was an incredible impact on reducing MS lesions. And so these are lesions that you can detect in MRI scans, which are associated with demyelination. And so in these trials, you essentially ablate the formation and observation of these lesions. And I was at the ECTRIMS meeting in Barcelona when this data was presented in 2015, and I was in a massive room. This was the keynote lecture. There's around 5,000 clinicians and scientists. And I'll never forget the gasp in the room when we saw this data, based on the fact that this was something that had never really been seen before, and we knew this was going to be a big deal for the field. So to summarize other results that were observed in these trials, in addition to this incredible effect on the formation of lesions in the context of depleting B cells, it was observed that there was a very robust and significant effect at reducing rates of relapse in relapsing remitting patients. So there's about a 50% improvement in reducing rates of relapse compared to the previous standard of care drugs. And in addition, it was demonstrated that this therapy resulted in statistically significant reductions in disease progression. And similar results were seen in the context of primary and progressive MS. And based on these collective results, the approval process was fast-tracked, and in 2017, this B-cell depleting antibody was approved for the treatment of relapsing, remitting, and primary progressive forms of disease. And so the question then is, why are B cells so important in MS? And how do they function in this context? And so a significant insight addressing these questions was provided by this comprehensive epidemiology study that was performed by Dr. Alberto Ashario at Harvard and his colleagues, which examined blood samples that were collected from a huge cohort of 10 million active US military adults, 1,000 of whom were diagnosed with MS during their service. And what this data allowed this group to identify was that unique amongst all the viruses that were examined, infection with EBV led to a 32-fold increase in risk to developing MS. And so this strongly suggests that EBV infection serves as a leading cause that triggers initiation of MS disease. And these collective findings led to the 2025 Breakthrough Prize being awarded to Dr. Stephen Hauser and Dr. Ashario just past weekend up in LA. And this is a very prestigious award. The ceremony is generally considered the Oscars of science. There's movie stars that present the awards. And as you may be aware, our own Jeff Kelly was awarded this prize in 2022, and I think it was Ed Norton that gave him this prize. But irregardless, the point is this is a significant breakthrough in the field of MS that has just really happened. And the final question is how, if Epstein-Barr virus causes MS, how is that, and what is the role of B cells? Well, it makes quite a bit of logical sense in that EBV virus primarily targets B cells. And upon infecting a B cell, it permanently transforms that cell, and so you have these lifelong B cells that reside in your body that harbor EBV virus. And so how does that infection lead to the development of MS disease? There's a couple of mechanisms that have been proposed. Following transformation of B cells with this virus, this leads to a redistribution of the capacity of B cells to go to the central nervous system, and that might be enough to get the initiation of an autoimmune response. Another very attractive hypothesis is that there's molecular mimicry, in that these B cells that are infected display an antigen, a viral antigen, which mimics a host antigen, and that leads to the priming of a T cell response. And so this is the hypothesis of molecular mimicry. And strong evidence for that particular hypothesis was published this same year in 2022. This was a big year in the field, in an article in Nature, where this group of researchers, again in San Francisco, profiled the blood of MS patients and then looked for cross-reactive antibodies that recognizes both EBV viral protein epitopes as well as self-epitopes. And they identified antibodies that were cross-reactive for both a transcription factor expressed by EBV and also a protein that's expressed on oligodendrocytes. And they pinpointed with molecular and atomic level resolution how you can achieve high affinity interactions with both of these epitopes. And this provides compelling evidence for the hypothesis that this is the origin of the initiation of autoimmunity in MS. And of course, all of these ideas never come out of a void or a vacuum. And in fact, our own Michael Oldstone had been talking about molecular mimicry as the origin of autoimmunity 40 years ago and was publishing papers in Science on the topic of how molecular mimicry between viruses and myelin basic protein and oligodendrocyte expressed protein could be the origin of autoimmunity. Again, the power and value of academic research in the development of new medicines. So just to summarize this first part of the talk, compelling recent evidence strongly suggests that EBV infection serves as a driver of MS disease and its ability to infect B cells. And this hypothesis is supported by the compelling and impressive data that's observed in MS patients when you deplete a B cell population. However, while there are statistically significant effects on disease progression with these particular therapies, unfortunately, even in the context of relapsing remitting MS disease, the disease does progress as patients age. And so there's, at present, a dire unmet need for the treatment of progressive forms of MS. And that brings us to the topic that's the title of the talk, which is targeting this regenerative process called remyelination, which, as I mentioned, has tremendous potential in the development of treatments for progressive MS. And so I probably said this about 15 times now, relapsing remitting versus primary progressive. What do I actually mean by this? MS disease can present in the clinic in one of multiple forms. It can present in what's called a relapsing remitting form of disease, and the majority of patients present with this form. Around 85% of new patients have RRMS. And then the other forms of disease are called progressive, and these are where you have no remission of disease, or you have secondary flares where you get decreased levels of remission. And without having time to go through the scientific data that suggests and supports this, disease remission in MS, where you have an inflammatory flare and then the disease resolves and you are basically symptom-free, is dependent on this regenerative process called remyelination. And so disease remission, this phase of remission, is dependent on your central nervous system having the capacity to go from the demyelinated axon back to this myelinated axon that can function, remyelination. This is a regenerative process that persists throughout adulthood in the central nervous system of mammals, of all mammals, and it involves and is dependent on an endogenous stem cell population called an oligodendrocyte progenitor cell, which is a highly abundant cell in your CNS. So about 4% of all cells in your brain and spinal cord are these OPCs, and they're broadly distributed and uniformly distributed. Highly abundant, present throughout life, and you have the capacity to remyelinate as you age. The process involves following a demyelination injury, so in the context of MS, a B-cell dependent T-cell mediated destruction of oligodendrocytes leading to demyelination. These stem cells become activated from their niche. They then migrate and proliferate, and then locally at the site of damage undergo differentiation to form a newly born oligodendrocyte, shown here in this light blue schematic. So injury leads to the activation, migration, and differentiation to form newly born oligodendrocytes, again at the site of lesion. And when you get this remyelination, that leads to functional recovery and disease remission. However, as this process begins to fail, this leads to progressive decline. And it's been clearly demonstrated that progressive forms of MS are associated with a failure of this regenerative process. Again, this beautiful model that I just told you about didn't come out of a void. This is 100 years of academic research. So it was first suggested in 1906 by a neurologist studying the disease that the spinal cords of MS patients had the capacity to remyelinate to facilitate disease remission. And it wasn't until 1979 that researchers clearly demonstrated that this remyelination process was actually functional. And so using models where you use a toxin to demyelinate neurons in the central nervous system, the spinal cord specifically, it was demonstrated that these lab animals had the capacity not only to remyelinate based on looking at the tissue with histology, but that the remyelination was actually functional. And so this leads to functional recovery. And then another major development in this field was the discovery and characterization of this stem cell population, the OPC, by Charles French Constant working in Martin Raff's lab in 1986, a number of years ago now. And so this was really opened up the entire field, that we now knew what the cell type was that you could potentially target for this. So then fast forward 40 years, we have this beautiful model. And again, I'm fast forwarding through 40 years of research, but I'm just going to highlight the two researchers that really developed this model, which are acutely intelligent and incredibly nice people. A guy named Robin Franklin, who did most of his work at University of Cambridge, who's now at Altos Labs, and Charles French Constant, who's the individual that identified OPCs from adults. And these two individuals have really pioneered the field and have enabled the drug discovery that now happens. Again, 40 years of research in an academic setting leads to a clinical hypothesis. Okay, so if we have this model and we have a hypothesis that if we target this process with a therapeutic agent, what's the best way to go about doing this, right? It's an incredibly complicated process. We know that the stem cell population is not limiting. If you look in the plaques of even aged MS patients, there's an abundance of these stem cells. But for unknown reasons, they either fail to activate or fail to differentiate. And so how are we going to modulate this, and what do we want to target specifically? So with any regenerative medicine-based approach, there's a number of initial considerations that you need to deeply consider before getting started. And so in the context of remyelination, is it OPC activation within an age niche, for example? Or is it differentiation at the site of injury, which becomes limiting, which you want to target with a therapeutic agent? The other question is, will a single molecule or mechanism work in all disease contexts? And so will something that works in a young person be the optimal therapeutic in the context of an aged patient? And also the consideration of different forms of disease here. And similarly, it's such a complicated process, will targeting a single mechanism or aspect lead to an optimal level of efficacy? And finally, I'm getting into the weeds here, but it's also important in the context of regenerative medicine of how you're going to treat patients with this therapeutic. So if you have something that's going to induce differentiation, do you have to worry about depleting the stem cell population if you treat with the drug chronically? So these are all things you need to think about and figure out as you go. Okay, so what I'm about to tell you is, to date, the majority of the efforts in this field, and this is the approach we took at the onset as well, was to target the differentiation of these OPCs within the lesion. And the rationale for this was that there was histology data in MS, I mentioned this, in the plaques of MS patients, where there was an abundance of these stem cells, but for unknown reasons, they failed to differentiate. But even within this one aspect, induced OPC differentiation, it's quite complicated. Okay? And that is because just going from the OPC state, assuming you've already activated the state, to the fully mature myelinating oligodendrocyte, there's at least one and probably two intermediate cell states that this OPC must pass through in order to get to the mature cell type. And each of these individual states is regulated by a complex network of molecular signals. And so this is a very complicated slide, and it's kind of on there. I have it up here for that reason. And when we started in this field, this is where we were. I said, okay, this is something that makes a lot of sense. You could target it therapeutically. The stem cell population is well defined, but how the heck are you going to do it, even if you decide you want to induce differentiation? And so this was a project that was actually optimal for this approach I mentioned at the beginning of my talk, which instead of having a specific target that you want to go after, we asked the question, could we use this phenotypic screening approach to identify molecules and mechanisms that get you from here to there? And so this is kind of the general theme of our research lab. We used unbiased, i.e. target agnostic, phenotype-based discovery, coupled with target identification studies to generate testable hypotheses. And so this is kind of the philosophy of the lab, is to use, instead of designing molecules to impact biology, letting nature's response to defined structures and mechanisms guide the generation of testable hypotheses. And the incredible thing about being at Scripps is, at this point, I went and talked to Pete and said, here's an idea, and he said, fantastic, here's the keys to the Ferrari, basically, because there's another place like Scripps to do this kind of research. And you know, this is, I'm saying, I'm going to take these stem cells I get from an optic nerve, and I'm going to screen 100,000 compounds using microscopy. Very few places on the earth you'll be able to do that, and especially not outside of an academic environment. So I'm very fortunate to be able to do this research. And so we did exactly this. We took these stem cells that we isolate from rat optic nerve. We can culture them ex vivo in a way that they retain their potential. And then working with an incredibly talented graduate student here at Scripps, a guy named Vishal Deshmukh, we developed an assay, a microscopy-based assay, where we simply plated these stem cells in a miniaturized dish, treated with compounds for 6 days, and then looked for compounds that induced the differentiation towards a mature oligodendrocyte fate, as read out by expression of this marker protein called MBP, which is associated with the mature cell state. And when we ran this screen, we identified multiple members of a class of known drugs that targeted a receptor called the M1 muscarinic receptor. So these were M1 muscarinic receptor antagonists. We identified around 14 compounds from this class that induced statistically significant induction of OPC differentiation. An example of which is this molecule called benztropine. And in collaboration with Rusty Gage's lab across the street at Salk, we found that the cells that the oligodendrocyte cells that we differentiated using these drugs were functional. Specifically, we showed in co-culture assays that these drugs would induce oligodendrocytes that could myelinate the axons of neurons that we co-cultured with them. So they were functional oligodendrocytes, which was incredibly exciting. And then working in collaboration with Ari Theophilopoulos and Brian Lawson, we showed that these molecules were functional in vivo in relevant models of demyelinating disease, both in the context of inflammatory-mediated demyelination and immune-independent demyelination. And without going through the details, importantly, we used electron microscopy to show that these molecules were working in this context by inducing functional myelination as opposed to preventing demyelination. So these were functional small molecule drugs, many of which were approved drugs that hit the target tissue in the central nervous system, which was quite an exciting finding for us. And we published these results a number of years ago now. What was really exciting was shortly after we published our findings, a lab at UCSF, Jonah Chen's lab, using a very elegant nanofiber-based assay where they looked for the ability of oligodendrocytes to myelinate a nanofiber in a 96-well plate, they converged on the same class of molecules, these M1 receptor antagonists. And so this is always exciting in academic research when two labs working completely independently and using orthogonal methods converge on the same answer or mechanism in this class. And this collective work led to this clinical hypothesis that antagonizing this receptor on OPCs would lead to functional remyelination in patients. And a clinical trial, an academic investigator-led clinical trial at UCSF, tested this hypothesis using clemastine as a tool compound. And impressively, this drug met its primary endpoint in this. And so there was evidence based on this assay called visual potential latency delay. There was statistically significant evidence for drug-induced remyelination. And this was exciting because this was the first demonstration of evidence for drug-induced remyelination in MS patients. And following the release of these results, 3 subsequent trials have initiated, 2 phase 2 trials and 1 phase 3 trial evaluating clemastine as a remyelinating drug. And one, just as kind of an aside, one exciting avenue and I think important avenue of research is combining remyelinating drugs with exercise. And so some people are, including in the clinic, exploring whether exercise enhances the function of remyelinating drugs. And this is because myelination occurs in an adaptive manner. Electrical signals on the axon actually stimulate myelination. So I think this is an important area of research. The other really exciting thing about this, besides being the first demonstration of drug-induced remyelination, which kind of provided rationale for the general hypothesis and approach, was that it demonstrated that the molecules and classes of molecules and mechanisms we identified using this phenotypic screening approach translated to human patients, which was incredibly exciting for us and kind of validated our platform for discovery. Now, so that was using phenotype-based discovery. I guess people that knew what they were doing, Charles French-Constant and Robin Franklin, took the opposite approach, where they used a target-based approach to developing a remyelinating drug. And so based on changes in expression of genes following demyelinating injury, this group of researchers discovered that RXR gamma, a gene called RXR gamma, plays an essential role as a positive regulator of remyelination. And Alistair Coles at the University of Cambridge, again an academic clinician scientist, tested this hypothesis using a repurposed cancer drug called bexarotene, which is an RXR gamma agonist. And in this study, using the same surrogate output for induced remyelination, they also saw a statistically significant change in latency delay, again suggesting that this molecule and target class could facilitate remyelination in vivo in human patients. And so this was really exciting. In a relatively short period of time, again, exclusively in the context of academic research settings, the field has progressed from a concept, so 40 years of academic research leading to a concept and hypothesis, to the identification of drugs that could be repurposed in the context of remyelination, all the way through to evidence for drug-induced remyelination in MS patients using 2 unique mechanisms. And so I think this is really exciting because I don't think there's another example of a regenerative therapy which has progressed this quickly. Now, unfortunately, there's issues related to toxicity for both bexarotene and clemastine. we think that we have addressed an issue with clemastine. So clemastine, I mentioned, the way it's working in this context is by antagonizing a receptor called the M1 muscarinic receptor. This is a drug that's an over-the-counter antihistamine drug, was developed to inhibit histamine receptor 1. The maximum concentration that you can achieve in a human is around 20 nanomolar, which is significantly lower than what's needed, we think, to induce differentiation. But by screening every known molecule that has annotated activity against this M1 receptor, we've identified an SAR and 2 candidates of approved drugs, which we think will be more effective than clemastine based on their safety profiles. So the other question that we've been very focused on of late in the last 5 to 7 years is this question of whether targeting a single mechanism in this context will allow you to achieve a maximal level of efficacy. And this question and serious concern actually came out of a serendipitous result that we observed working with Gary's lab here at Scripps, where we had a hypothesis that metabolism plays a critical role in the maturation of oligodendrocytes. And so we were doing this exercise that Gary calls metabolite activity screening, where we look for all differentially regulated metabolites and metabolic pathways associated with 2 distinct cell states, here looking at the OPC and mature oligo state, and then fed back those metabolites to see if they impacted the process on their own. And what we observed here, again, without having time to get into the detail, is that if you add an aminosulfonic acid, a metabolite called taurine, to OPC differentiation, you have this incredible impact on the overall efficacy of drug-induced differentiation. And so if you add taurine to the media of any drug-induced differentiation, you achieve this incredible level of efficacy. And so whether you can modulate taurine levels in vivo in MS in a relevant way, and whether this is actually relevant in the context of whole animal physiology is a question. But what this made us realize was that if this was the signal, the maximal signal you could achieve with drugs like clemastine in the clinic, what would you achieve if you had combinations of drugs that gave you this kind of an output? And so this was the kind of eureka moment of saying, okay, we've achieved that first step, but we think we can do much better. And we think these combinations make perfect sense. Again, you're asking a lot of a single mechanism to get you all the way from here to there. We think the way that taurine combination works is that the one mechanism allows the OPC to exit the cell cycle, and then taurine it facilitates maturation. But irregardless, at this point, we sought to say, are there known drugs that we can combine with these clinically validated mechanisms to achieve a maximal level of efficacy? And this is the work of a very talented graduate student in my lab who graduated a few years ago, Brittany Beyer, who asked the question, can we improve the efficacy of drug-induced remyelination by identifying optimal and developable drug pairs? And the way she did this was she did combinatorial drug screening, where now, instead of looking at a single drug, we're looking at matrices of an 8x8 matrix of different drugs and looking for additive or synergistic effects. And I'll summarize 5 years of grueling work in 2 slides here, but she found 2 combinations that we think are pretty interesting. The first was if you combine a vitamin D receptor agonist with drugs from this M1 receptor antagonist class. So for example, if you add vitamin D or analogs thereof to clemastine-induced differentiation, you get this additive effect at induced differentiation. And we also found that vitamin D is a very complicated vitamin. It needs to be metabolized in your liver and kidney. But what we found is that OPCs actually have the capacity to metabolize vitamin D precursors, which suggests that this particular mechanism is really important to OPC physiology. This is potentially also interesting because vitamin D has been described in the MS literature for years, in that there's this hypothesis that one of the reasons why there are these geographical differences in incidence rates of MS disease is because there's differences in the amount of sunlight that different people get. And so vitamin D, you either get it from your diet or the precursor is made in your skin following exposure to UV light in the sunlight. So your vitamin D levels are correlated with sun exposure. And this had been proposed in the literature for decades, but again, this Alberto Achario, who I just told you about, who won the Breakthrough Prize for elucidating the role of EBV, actually ran a clinical study and demonstrated that the conversion to fully diagnosed MS directly correlates with levels of vitamin D. And that led to a clinical trial, which was just actually read out and released very recently, where this group of academic clinicians in France evaluated high-dose vitamin D in the context of, again, progression from an initial diagnosis to clinically defined MS. And in fact, they observed a significant reduction in conversion to clinically defined MS disease in the context of high-dose vitamin D. What these results mean, I don't know, but we are confident based on our observations that vitamin D signaling plays an essential role in OPC differentiation. The other interesting combination that Brittany identified was the observation if you combine bexarotene, so that was the mechanism that the Cambridge group identified, with small molecules that inhibit enzymes associated with cholesterol biosynthesis, including drugs like tamoxifen, which is how it's working in this case. You see a synergistic effect at drug-induced differentiation. And we think this is important because, as I mentioned, bexarotene had dose-limiting toxicity. And what we see is if you combine bexarotene with tamoxifen, you get not only a synergistic effect at induced differentiation and efficacy, but you're also dose-sparing with respect to bexarotene. And so we think you'll be able to use a lower dose of bexarotene in the clinic. And so just to summarize this later part of the talk, we've identified a second generation of M1 receptor antagonists, which we think will be more efficacious than clemastine. And we have two clinical hypotheses that we would love to test in the clinic, these two particular drug combinations. Okay, so just with the final 6 minutes I have here, I just want to tell you about some of our thinking and our current working addressing this last question of whether a single mechanism will work in all disease contexts. And kind of a primary area of focus here, not only for us, but the entire field, is an understanding that this regenerative process of remyelination and the capacity of your spinal cord and brain to regenerate is very clearly age-dependent. And so it's been demonstrated both in animal models and also in observations based on postmortem tissue of human patients that your capacity for remyelination deteriorates as a function of age. And if you look at the responses of MS drugs, specifically here looking at response rates to bexarotene as a remyelinating therapy, there's an inverse correlation of response as a function of age. And this is not only true in the context of remyelinating drugs like bexarotene, but most MS-related drugs begin to fail as you age. What's been established is that this loss of remyelination capacity as a function of age is not the result of an inherent limitation of the OPC stem cell population or a loss of those stem cells. So in aged tissues, both in rodents and in humans, there's still an abundance of these OPCs, but for unknown reasons, they fail to activate. And a really interesting area of trying to reactivate these aged OPCs is associated with something that is kind of near and dear to our heart here at Scripps. And so again, Robin Franklin's lab was studying mechanisms that were responsible for inhibiting OPC activation in an age niche. And what they realized, which is consistent not just with the OPC niche, but within CNS in general, is that as you age, your tissue becomes harder. And this was made clear to me when I visited Cambridge, and the postdoc that was working on this showed me the brain of a young versus a geriatric mouse. And the consistency of the tissue was similar to kind of cream cheese just as you open the package and then after it's been left open for a couple of weeks. Not kidding. This is the reality. And you can measure and quantify this using techniques like atomic force microscopy. And they demonstrated that this kind of general observation is true and that there's a significant stiffening of the matrix of the central nervous system as a function of age. And what Robin's lab showed is that if you take aged OPCs out of the animal and then culture them on a matrix that mimics the stiffness of a young animal, the cells had the capacity to differentiate, which suggests that this mechanosensing plays an important role in the inhibition of OPC activation. And incredibly, they showed that if you knock out piezo-1, the mechanosensor piezo-1, specifically in OPCs, you restore the capacity of these cells to remyelinate in aged animals. So they're taking a year-old animal, they're specifically knocking out or knocking down piezo-1 in OPCs and showed that you have a significant enhancement in the capacity for these cells to become activated. And this is really exciting because this is, as you may be aware, if you came to Ardem's seminar, I'm sure you're aware of Ardem's research. Ardem won the Nobel Prize for his work discovering piezo-1 as a mechanosensor, piezo-1 and piezo-2. And I'm sure, I don't know, Ardem, did you ever think that piezo-1 would play an essential role in regulating stem cell activation in the context of aging? Of course, yeah. But again, the value of academic science, right? This is a discovery that now has, we think, significant implications, and because this could be significant not just in the context of OPCs and remyelination, but multiple stem cell related mechanisms could be inhibited in the context of aging for this reason. And this is my last slide. I mentioned this concept of activity-based myelination, what's called adaptive myelination. So everything I've been talking about so far has been in the context of disease, which I guess can be somewhat depressing, but it's really an exciting time to be a myelin scientist. There's an emerging literature which clearly is demonstrating the essential role of OPC differentiation and myelination in contexts like experience-dependent learning and motor function. And so potential applications of the types of drugs we're identifying is to explore some of these concepts in vivo, which we hope might have utility in kind of context of basic biology. Okay, so to summarize, I told you today about the exciting new developments in our understanding of the molecular basis of MS disease and the role of EBV, viral infection of B cells as a potential primary driver of MS. And I told you about the incredible efficacy of these repurposed B-cell depleting antibodies in the context of relapsing remitting forms of disease. Now, targeting your B cells, these antibodies were developed in the context of B-cell related cancers, is a pretty serious thing to do. So you're depleting a significant arm of your adaptive immune response, which has potential safety issues. And if there are multiple second-generation approaches that are going forward now, one of which is doing things like using CAR T cells. And if you're interested in this, this is a vignette from the Scripps webpage today where my friend and colleague Travis Young just got FDA approval to try his switchable CAR T cells in the context of autoimmune disease. I also told you about the complete lack of drugs for progressive forms of MS and the potential we think that remyelination promoting drugs might have in this particular area. And I told you about what we're calling next generation muscarinic receptor antagonists and these two clinical hypotheses we would love to test now pending funding. And so Cambridge actually has an approved clinical protocol for testing this hypothesis. We just need to get the funding to do it. So we're very excited about that. And then finally, I tried to do this as I was going along, but just as a take-home, I showed these faces as I presented different areas of research, trying to highlight that all of this was done in the context of an academic research lab. So Steve Hauser had the volition to try and repurpose B-cell depleting antibodies. Epidemiologists at Harvard elucidated the potential role of Epstein-Barr virus. These findings don't come out of a void. The entire field of remyelination was pioneered by academic research like these two individuals. And people like Michael Oldstone were pioneering and providing this hypothesis of molecular mimicry 40 years ago. So with that, I will thank everyone in the lab that has done the work. I'm just highlighting people here, the current lab, as well as past members that have contributed significantly to this work. An incredible group of collaborators at Scripps. As I'm starting my talk, I'm talking to all these faculty members that I've got collaborations going with, which is phenomenal. And I especially have to thank our funding bodies. CIRM provided the majority of the funding for this project. And I also want to highlight contributions from the David C. Copley Foundation, which actually provided a very timely infusion of very necessary funds that allowed this research to progress. So with that, I'll close and I'll take any questions in the forum. Thank you.
Great. Okay, thanks. Join me, Luke. So wow, that was remarkable, and I come away with this sense of wonder about everything is connected.
All right.
And so I'm going to start. I have a interesting question from the virtual audience, and you showed great evidence that vitamin D, exposure latitude kind of explains incidence of disease. And other metabolic effects contribute. Yep. And other metabolic effects. And one of the things that you mentioned in your intro, but you didn't come back to, was women.
Right.
So what do you have any further insights into why women are more susceptible, and how do all of these remarkable kind of effects that you found bear on that?
Well, I'm not. I'll speculate here, but I mean, like many autoimmune diseases, this is an observation, right? There's a phenomenon that women are more susceptible to many autoimmune forms of autoimmune disease. And, it probably just equates to differences in gene dosage expression. And one really important example, which my colleague Alejandra made me aware of recently, is that the key gene associated for the differentiation and production of the T cell population that's responsible for suppressing immune response is actually X-linked.
Okay.
And so these gene dosage in X-linked dependent fashion probably contributes to differences in immune responses that makes females, members of the female population, more sensitive to that triggering that leads to breaking tolerance and autoimmunity.
So
I'm speculating.
That's a hypothesis?
Hypothesis,
Needs to get borne out.
Yeah.
Okay, now if you have a question in the real audience, raise your hand and somebody will come around with a lightsaber so that I can see. And in the meantime, I'm going to ask a question, and I'm veering to the edge of my knowledge here, but I was curious why you got muscarinic acid receptor, antagonists as hits in your screen. Right. So that's like a neurotransmitter.
The mechanism?
Yeah. So what is? So that's like the wiring, not the insulation.
Right. Yeah. So
is that fair?
I mean, I didn't have time to get into it, but there's an entire controversy in the field as to the mechanism of action for that particular class of drugs. We're very convinced, and we published it in our paper, that class of drugs impacts calcium import in OPCs, which is what M1 does, and there's a body of literature of the role of calcium signaling in the excitive cell cycle of OPCs. There's compelling evidence to suggest that there's at least one other mechanism that's required for that class of drugs to work. So, and I honestly believe that it's polypharmacology in this class. Okay. Yeah. And so the key question is, if a drug from this class can do it, is it going to be safe in a human patient? And we're working to understand the mechanism.
So we have here's one down in front.
That was really interesting. Thank you. I was really interested when you were talking about the matrix stiffness and the effect that has. Is targeting the stiffness of the matrix a potential mechanism that could be used alongside these other approaches to improve efficacy? How viable is that?
Yeah, no, absolutely. I mean, and this is actually, if you're interested, this is one of the grad students in the lab, Corey, this is one of his main projects, is to see if we can identify molecules that modulate that process. So we're taking, in this case, we're taking young OPCs, culturing them on a stiff matrix, and we see inhibition of differentiation, and then looking for molecules that overcome that mechanism. Direct antagonist of piezo-1 is kind of a holy grail in the field, which I think people on this side of the room are very actively working on. But that would be a potential application for those drugs down the road, for sure.
Yeah, another one down here.
Yeah, very, very nice. You presented a number of examples where, from an academic environment, the things were actually tested in humans in clinical trials. So, from your own work, how do you decide when it's time for your results to be tested in humans? And maybe you've already decided that. And if so, what are the barriers to doing that?
Yeah, I mean, so this is the big issue with phenotypic screening, and why the industry has kind of soured on it, is that there's a large step in going from identifying a molecule that does something in a cell, even in vivo, to understanding the mechanism of action, which is absolutely required before you could test it in a clinical setting, right? And so the advantage of the first class of molecules we published, and the reason that we really use those as tools compounds to kind of set the foundation, was because these were approved drugs that had known human safety profiles and were known to distribute to the target tissue in the brain, which is a big issue. And so that allowed the immediate translation of those findings. But for a small molecule that has an unknown mechanism of action, it's definitely a longer road. And the answer is you want to know the mechanism of action, have a complete confidence in that target, genetic validation of the target you've identified, and then an understanding that you can achieve a safe level of that drug in the target tissue.
A question up here.
Yes, thank you. Steve Clary here from the Osher Lifelong Learning Institute at UC San Diego. Many of your colleagues have been speakers. Intriguing about the BSV virus. If it is considered to be a precursor of MS, are we not looking at vaccines and things like that? And where does the virus come from? You're the 10 million service members with a 992 possessing the virus is rather intriguing.
Yeah, I mean, this is a, I mean, yeah, how do you dig through that data to come to that conclusion that EBV causes MS? Because it's something like 99% of all people will develop or will be exposed to Epstein-Barr virus at some point in their life, right? So that's the question. But that's, you know, by looking at serotype matching before and after induction of MS, that was the only virus that you had that observation. But you're absolutely right. I mean, all of these therapies where you're depleting B cells, which are effective at blocking the immune response and decreasing rates of relapse, they're definitely not a cure, right? If you still have those EBV infected cells, how long do you have to treat with those therapies? So for sure, having an EBV vaccine that's efficacious would be a huge thing that you, based on this hypothesis, you would predict would significantly reduce rates of MS disease around the world. And so, I know, Andrew, where are we with a Epstein virus vaccine? Yeah, yeah. No, that's the solution. Is if that's really is an effective vaccine for EBV, but that's definitely not my area of expertise or research.
Great. We have another one down here.
Hey, great talk. I was wondering, two-part question really. But so first, by targeting M1R and RXRG, you can revert progressive forms of MS to RRMS, or basically remyelinate, which is a feature of RRMS, right? And in that case, can you then further treat it by applying B-cell therapy? Because we know that B-cell therapy can help alleviate RRMS symptoms. And then, so that's question one, I guess. And then question two is, have you ever tried to see how much, like, because M1R and RXRG are two different receptor pathways. Mm-hmm. Have you ever tried combining both drugs and then seeing like whether that plus B-cell therapy or whatever can eventually, like how much recovery that can give to a mouse model or a human model?
Yeah, so I mean, the answer to your first question is I'm for sure not proposing that these drugs will eliminate the need for suppressing the immune response. So these are complementary therapies that would be used to deal with progression. But for sure, you're going to want to block the immune response. And what Robin always says is you're not going to rebuild the house while it's still on fire. So you've got to block the inflammation and inflammatory response. So for sure, these would be combination-based therapies in that context. Your second question, we definitely tried every possible combination of agents. So and uniquely, we found those two combinations I told you about, the Bex plus TAM and the M1R plus vitamin D receptor. So the answer is you don't see anything when you combine those two.
Okay. Oh, we have another one. Yeah, good.
So one of them you answered, that is a combination therapy, because the more of myelin you create, the more it becomes like an attractive target for the immune system to attack. So obviously a combination therapy where you're trying to blunt the immune response to the self, plus trying to generate more cover for the neurons would be great. So on the same topic of the vaccine, has anyone tried like an antiviral drug for EBV in these patients and seeing that they respond in a much better manner to the treatment?
Well, the tricky thing there is basically you would have to give children and feed them prophylactically with that antiviral drug to prevent the infection, because you couldn't treat it retrospectively, right? The EBV infection, you'd need to prevent the infection, I think.
Yeah, I think so. Yeah. So something, so let me put a point on one of your messages, which is that, you made a really compelling case for academic medicine and all of this. So, there would be no drugs coming out for MS unless people like you were doing this. And all of the funding that you got was from foundations and the federal government. Right. And I think maybe we can take the onus of saying we haven't really, as scientists, haven't communicated effectively to the general public how crucial it is to support all of this. And it's, drug companies can't do this kind of research.
Yeah, they're not going to do it.
Because sometimes stuff you do doesn't work, and they need to have a profit. So they can only enter
at the validated target stage.
At the validated target stage, and somebody has to figure all this out. And this is like a 100-year story to figure this out.
Yeah.
So I hope everybody can appreciate the role of government funding at a very large scale, which all around the country supports research like this that creates the medicines for tomorrow. Yeah, well, okay. Say that again. Okay, so on that note, I want to thank you all for coming and thank Luke for a great presentation. Now his whole lab is here, and they've set up a demonstration out in the back. There's science in action, and you can peer through a microscope at some neurons. There's a couple of other demonstrations, little robots. And there's a reception for those of you who are here, but the last thing I have to do is point out that we have the next front row. I think it's the last one for this season. So we have Eric Topol, who's a very special communicator of science, and I think it'll be a terrific lecture. That's May 21st. So I encourage you to log in by Zoom, but I'd also encourage you to come here and attend in person. And if you do, you'll find me in the front row.
Thanks, man.